Hello. Hi, I'm Dr. Hilary Michelson, and I'm president of the Robert F. Furchgott Society. And on behalf of myself and Dr. Danny Rosenbaum, who I know is here somewhere, the vice president of the society, I am so happy to welcome all of you to the 2018 Visiting Professor Lecture. The Furchgott Society, as many of you know, was established in 2005 to honor the legacy of Dr. Robert Furchgott, who received the Nobel Prize in 1998 for his contribution to the discovery of nitric oxide as a signaling molecule in the cardiovascular system. Discovery, as we all know, that's had a wide-ranging effect on clinical medicine and biomedical research as well. This year marks the 20th anniversary of Dr. Furchgott receiving the Nobel Prize, and we are so honored that our visiting professor this year is a co-winner of the award with Dr. Furchgott, Dr. Lou Ignarro. It's my honor to introduce the president of SUNY Downstate, Dr. Wayne Riley, to offer a few remarks.
Well, good afternoon. What a great week here at Downstate as we mark the 20th anniversary of our beloved late professor, distinguished professor, Dr. Robert F. Furchgott's co-receiving the Nobel Prize in Physiology and Medicine. And you heard me say, some of you heard me say this yesterday. When I got here in April of 2017, it was within a couple weeks that I had a conversation with Rose Jackman, and I said, Rose, where's the Nobel Prize? She said, oh, it's in the safe. And I said, my gosh, we got to get that thing out of the safe and put it on display, because I wanted the students, the faculty, visitors to see the great work that happened within, quote unquote, the four walls of this great institution by Dr. Furchgott and his co-investigators. Because there's not many institutions that you go to that have had a Nobel Prize winner in physiology and medicine, and I wanted it to be a daily reminder of the great work and our great calling as biomedical scientists, physicians, residents, and students to be inspired by it when you walk by it. And just after 2 days, Dr. Ignarro, we've only had the unveiling. So yesterday I was leaving to go to Manhattan for a meeting, and I was just tickled that people were standing there looking at it and trying to read the proclamation in Swedish. But that's exactly what my intention was, colleagues, that we put this mark of achievement that means so much to scientists all over the world, and that we honor Dr. Furchgott by doing that in this great week, and the exhibits and the dinner tonight. And more importantly, today, the co-recipient, one of his two co-recipients, the distinguished Professor Ignarro, yet another proof that good things come from Brooklyn. He is Brooklyn born. You know, I haven't added it up, but I suspect that Brooklyn has probably had a pretty good number of Nobel Prize winners. And, you know, in a lead up to this, this is a God's honest true story, only in New York. About a month ago, I was in Grand Central Station. It was about 10 o'clock at night. I was waiting to get on a train to go to Albany for a meeting at SUNY Central. And remember, during the summer, they move all the Amtrak trains to Grand Central from Penn Station. And I'm just waiting there, and guess who walks by is another Nobel Prize winner in medicine, and that's Dr. Harold Varmus, who I knew from when I chaired the NIH panel on minority health and health disparities. And I said, Harold, where are you going? He said, Wayne, I'm going to a meeting. Where are you going? We were just chatting. So it just struck me. I'm chatting with another Nobel Prize winner in Grand Central Station at 10 o'clock on a hot summer night. And I said, Harold, you've got to come to the festivities that we're having to honor Dr. Furchgott. We're having Dr. Ignarro come by. And he said, oh, Wayne, just please tell Dr. Ignarro I have a conflict to be out of the country, but I would be there if I can. So I bring you greetings from Grand Central Station in the middle of the summer from another Nobel Prize winner. So again, colleagues, this is a great, great event for a great, great man. And the more and more I learn about Dr. Furchgott, he and I share that we're both Southerners, born in South Carolina, Charleston, lived his early years in Orangeburg, South Carolina. And he lived in the Deep South, or the South that was pretty segregated. And he used to talk about that, I understand, with his colleagues. And he was very committed to South Carolina and making sure that kids in South Carolina had the same opportunities that any other kid anywhere in the country had. So another tidbit about our late great colleague that I think really is emblematic of the man and emblematic of the moment that we celebrate the 20th anniversary of he, Dr. Ignarro, and Dr. Murad being awarded that great prize. So on behalf of all of us, Dr. Ignarro, welcome home to Brooklyn, and thank you so much, and thank you for your great work and the honor of a lifetime that you shared with Dr. Furchgott. Thank you.
Part of the mission of the Furchgott Society, besides bringing such prominent and world-renowned scientists to our campus for all of us to become inspired by, is to provide grants and awards to promising young investigators at downstate. To date, I think, in great part due to this, the generosity of our donors, sponsors, and the members of the Furchgott Society, and if you're not a member, now's the time, I encourage you all. To date, again, due to the generosity of our supporters, I think the Furchgott Society has raised close to $500,000 that we've been able to share with the downstate community through grants, awards, and other sponsored events such as this talk. And I'm so proud of our society for being such an important contributor to the downstate environment. I'd like to introduce now Drs. Todd Sachter and Jeremy Copeland, who I don't know if Jeremy is here. They served as the chair of the basic science review committees and the clinical science review committees for the Furchgott Society. And they assessed all the qualified applicants and are going to announce the recipients of the Furchgott Scholar Awards. So please welcome Dr. Sachter, and I guess I'll stand in for Dr. Copeland.
So the Robert Furchgott Scholar Award is given to a postgraduate scientist whose mentored research fits best with, and it is inspired by the research of Dr. Furchgott. And so this year's winner is Irina Abayeva. Please come up. So Irina did her work in Chris Helen and Tatiana Pestova's lab, and the work really involves the mechanism by which viruses, when they infect cells, can take over the protein synthesis machinery of those cells. But the genetic material, the RNA or the DNA of the virus, is so small that when it takes over that mechanism of protein synthesis in the cell, it reveals the mechanisms of protein synthesis in ways that we would never have understood. So what Irina's work was on the very smallest unit of a RNA virus that could take over the cell's protein machinery. And what she showed is that it actually is so elegantly short circuits the protein synthesis machinery and takes it over in a way that had never been seen before. And so it is in the spirit of Dr. Furchgott's research, because Irina's research not only reveals something fundamental about biology, but may also one day lead to new viral, antiviral therapies, and also might reveal how the RNA world, in the beginning of life on Earth, could make proteins, which is really one of the outstanding mysteries in science today. So, please.
I actually am so sorry because Dr. Jeremy Copeland was the chair of the Clinical Sciences Review Committee for the Furchgott Society. So I'm going to do, not such a good stand in an introduction. So I apologize to Andrew, beforehand. But I do want to introduce the Furchgott Scholar Award winner for the Clinical Sciences Division, and that's Andrew Malamus. So Malamus. And I practiced, but I still. And Andrew is a resident in the Department of Dermatology. He did his work with Dr. Jared Jagdeo. Did I pronounce that correctly too? Thank you. And again, I'm not going to do his research justice. Oh, yes, saved by the bell. Dr. Copeland, please join us on stage so you can do a much better introduction of our Furchgott Scholar Award winner. Thank you. Timing is everything in this world.
Sorry, I didn't realize first up. Hello, everybody. Welcome to SUNY Downstairs. Welcome to our speaker today. And my name is Jeremy Copeland, and I'll be presenting the award for the best resident or fellow. We had some excellent presentations this year, and I'd like to present Andrew Mamalis with his award. He's right over here. Come up, sir. Here's your check, and here's the plaque as well. Okay, all right, very good. Well done.
We're a very informal and supportive society and inclusive. Please join. Okay. I last would like to introduce the student who's going to actually do the introduction for our speaker. Can I please call up to the stage Abi? I'm going to pronounce this one correctly too. Amranani. I was close, right? Thank you. I've been working. So who's going to do the introduction for our visiting professor, Dr. Lou Ignarro. Thank you.
Hello, everybody. Thank you for being here. So I'm proud today to introduce our 2018 Furchgott Visiting Professor Lecturer, Dr. Lou Ignarro. A native Brooklynite, he completed his doctorate training at the University of Minnesota, finished his postdoctoral fellowship at the NIH, and then began his Nobel Prize winning research into nitric oxide and its relationship to cyclic GMP at Tulane University and the University of California, Los Angeles. His numerous discoveries, including that nitric oxide induces relaxation of vascular smooth muscle, has led to many clinical advancements, including the use of nitroglycerin in acute coronary syndromes. Beyond his many accolades, which you can read in detail in the packet you have from today, Dr. Ignarro is also a passionate educator of undergraduates, medical students, graduate students, and developing scientists everywhere. In fact, for his teaching at UCLA, he was awarded the Apple Teaching Award by first and second year medical students at UCLA 11 years in a row. And today, some of us downstate students were also fortunate to experience his passion for teaching. We learned about his time growing up in New York, including his accordion lessons that he took here in Brooklyn, and how Dr. Ignarro developed his trajectory to become the preeminent scientist, scholar, and educator in his field. So now let's give a warm welcome to Dr. Lou Ignarro as he shares with us his highlights in his presentation titled The Road to Stockholm.
Thanks very much, Avi. That was a very nice introduction. I want to thank you. I want to thank also Miss Rose Jackman for organizing this event, especially my travels and dealing with all of my emails. Thanks, Hillary, for all the kind words and pronunciation of all the names. And Dr. Riley, who I think had to leave for an important meeting. That's how deans are these days, you know. It's really great to be here at SUNY Downstate Medical Center to help celebrate this 20-year anniversary of the Nobel Prize for nitric oxide. And it's also a great honor and a pleasure to be here because this is the place where my good friend Robert Furchgott was for so many years. And so Professor Robert Furchgott was a colleague of mine, also a good friend of mine for many, many years. We first met at a pharmacology meeting in 1964 when I was a graduate student, and that's about 54 years ago. Bob was working on drug receptor interactions and kinetics, and I was working on catecholamines in the developing embryonic heart. And when I graduated from the University of Minnesota, a Department of Pharmacology, in '66 with a PhD in pharmacology, I was fortunate to have published 4 back-to-back papers in JPET, Journal of Pharmacology and Experimental Therapeutics, apparently a record that still holds. I wasn't after any records. I just wanted to get my degree and get the hell out of the cold. It was so cold in Minneapolis. But Bob Furchgott was quite impressed with these publications, and he offered me a postdoctoral position in his laboratory right here. However, I did not accept the position because I wanted to continue my training in chemistry and chemical pharmacology. I had a good physiology background, but I had a passion for chemistry and a chemical input to pharmacology, which was not that popular in those days. And so I accepted a postdoc at the newly created Laboratory of Chemical Pharmacology at the NIH in Bethesda. So who knows what might have happened had I joined Professor Furchgott here in Brooklyn. But I think if he were here, he would join me in saying that we're glad things turned out the way they did. And we remained good friends from that point on. Bob's basic research and mine brought us even closer together in 1980 when he discovered EDRF. His work and my work culminated in the both of us sitting next to each other on a stage in Stockholm in 1998. I mean, who would have ever thought? And there he is sitting right in the middle. You can always spot me immediately by looking for the change in reflected light right over my head. Here's Bob, here I am, and this is Fred Murad, and I'll get back to that slide a little bit later. And as I said before, this is the 20th anniversary since the Nobel Prize. In fact, this month, October, 20 years ago, is when the Nobel Prize was first announced. Now, my undergraduate studies were in pharmacy and chemistry at nearby Columbia University. And because of my passion for both biology and chemistry, I decided I wanted to do graduate studies in pharmacology, which is almost by definition physiology or biology and chemistry. And then I did a postdoc, as I said, in chemical pharmacology. Now, for the first 10 years of my research, I have to say my interests were all over the place and were not at all focused. I went from graduate studies on catecholamine synthesis, storage, and release in the heart. I was following what Julius Axelrod was doing at the NIH. And as a postdoc, I worked on the characterization of beta-adrenergic receptors, did not make much progress there. And then I went into drug industry, working on the development of novel anti-inflammatory drugs. But that only lasted, industry lasted for only 5 years, because I wanted to go back to an academic setting because I knew that I just wanted to teach students, medical students, graduate students, undergraduate students. Also, I wanted a bit more freedom in the kind of research that I was doing. And so, upon starting as an assistant professor at Tulane Medical School in New Orleans, my research shifted again, but this time for good. I got interested in cyclic nucleotides, particularly cyclic GMP, because very little was known about this molecule. A lot was known about cyclic AMP, but virtually nothing about cyclic GMP. And while studying the pharmacology and the biosynthesis of cyclic GMP by guanylate cyclase, something fascinating happened. The atmospheric gas nitric oxide was reported to activate guanylate cyclase, the soluble isoform of guanylate cyclase, thereby elevating tissue levels of cyclic GMP. And as shown in this particular cartoon, this discovery was made by Fred Murad, and with that discovery, Murad created and launched the field of nitric oxide research. Now, unlike many of you, unlike all of you perhaps, I'm no longer active in the laboratory setting. I've retired about 5 years ago, and I'm loving it, just in case you want to ask me how I feel. And so I can't present any new observations, and I don't have 3 or 4 hours to review the field of nitric oxide. So my plan is to tell you instead some of my own early thoughts on how I addressed various questions as they came up in order to answer certain key questions and better understand a most unique signaling molecule, nitric oxide. I mean, I really believe that nitric oxide is truly remarkable. And the more I do in this field and the more I read about this field, the more I'm convinced that it's a truly remarkable molecule. Very simple molecule, as you know, 1 atom of nitrogen, 1 atom of oxygen. It's paramagnetic, that is, it's a free radical. It has 1 electron that's delocalized or shared between the nitrogen and the oxygen atoms. And of course, this reactivity accounts for its biological activity. We began studying NO in about 1978, just after Fred Murad showed that NO activates guanylate cyclase. We had been studying guanylate cyclase for several years before the discovery of NO, just because I like to study enzymes and enzymology. But now we wanted to understand how NO activates guanylate cyclase. What is the mechanism by which a free radical can activate an enzyme protein? We were also interested in learning more about the mechanism by which the NO cyclic GMP system relaxes vascular smooth muscle. Now, at the time, we viewed nitric oxide purely as a chemical. It was not known to exist in cells. I mean, we viewed it as a dangerous, highly unstable gas that rapidly oxidizes in the presence of oxygen or air to nitrogen dioxide gas, or NO2, which, if inhaled, could kill you. And believe me, I experienced just that one morning in the laboratory, which had a dysfunctional fume hood. But fortunately, all that happened to me is that I lost my hair. The rest was okay. So cyclic GMP, but not cyclic AMP, in tissues was markedly elevated by nitric oxide and by certain drugs that contain NO or release or generate NO. For example, NO donor chemicals. Fred Murad showed this, and a good example is nitroglycerin. So nitroglycerin was known to be a vasodilator since the 1870s. It was actually discovered in Alfred Nobel's dynamite factories in Stockholm in the 1870s. Nitroglycerin is a volatile oil and gets into the air. Of course, Alfred Nobel used nitroglycerin as the active explosive in his invention of dynamite. When workers would come into the laboratory on Monday mornings, they would all get headaches because of the cerebral artery vasodilation, but some of the people also suffered from angina, and when they went to work in the factory, their chest pains and arm pains would disappear for the whole week. And then on the weekend, when they were away from the nitroglycerin fumes, the pain would come back, sometimes with very bad consequences. So the physicians in the community quickly realized what was going on, and they very carefully took some nitroglycerin, tiny amounts, and developed small triturated tablets, which they discovered if you place under the tongue, works in 20 or 40 seconds. And of course, we still use those tablets of nitroglycerin today. But the mechanism by which nitroglycerin works as a vasodilator remained unknown for over 100 years. And we thought, as did others, could it work by a nitric oxide, a cyclic GMP mechanism? And so I thought it was time that we tried to figure this out. Early on, we were puzzled by, but yet intrigued by certain observations, many of which were not published. You know, you don't publish every single thing you do in the experiments, but later on, it's fun to look back and see, you know, what you thought or what were in your laboratory notebooks. And I'd like to just share some of this with you. For example, we found that although nitroglycerin activates crude preparations, unpurified preparations of the enzyme guanylate cyclase, it couldn't activate the purified enzyme, and we didn't know why. When we added back some crude homogenate supernatant fluid, then it restored, then it allowed nitroglycerin to activate the purified enzyme. So we thought that perhaps nitroglycerin required a cofactor to activate the enzyme, and maybe this cofactor was important in vivo, you know, for nitroglycerin metabolism to nitric oxide. And so we set out to determine whether nitroglycerin might be metabolized in vascular smooth muscle. And this is what we found doing a variety of experiments, which I'm not going to share with you, but we found that when we incubated nitroglycerin with vascular smooth muscle preparations under various conditions, nitroglycerin was converted to nitric oxide and also the dinitro alcohol. When we tested the dinitro alcohol, it was inactive as a vasorelaxant, so the relaxation of the artery, the arterial smooth muscle, was due entirely to nitric oxide. And so how did this occur? In other words, how was nitroglycerin converted to nitric oxide? So we observed under a variety of conditions that when we added the thiol amino acid cysteine to these preparations, the enzyme preparations, the cysteine restored the capacity of nitroglycerin to activate guanylate cyclase. So in the purified enzyme system, nitroglycerin would not activate. When we added back either a homogenate supernatant or cysteine, then we can get nitroglycerin to activate the enzyme. And there was a concomitant formation of something called S-nitrosocysteine, an S-nitrosothiol. The S-nitrosothiol was red in color, it was fairly unstable, it markedly activated guanylate cyclase, and it did so because it was an NO donor chemical. In other words, the S-nitrosocysteine spontaneously broke down to release NO, and so the NO caused vascular smooth muscle relaxation. In doing further experiments, when we incubated the nitroglycerin with vascular smooth muscle, we found that nitrite, or NO2-, was generated from one of the two organic nitrate ester groups, and subsequently that nitrite could react with cysteine to form S-nitrosocysteine. We identified that. The S-nitrosocysteine works as a vasodilator by spontaneously breaking down and releasing NO. And many years later, Jonathan Stamler, who's a very productive pharmacologist these days, showed that mitochondrial aldehyde dehydrogenase actually catalyzes some of these reactions that I've been talking about. And so if I go back to S-nitrosothiols, we synthesized and tested about 10 different ones. 4 are listed here. They're all good NO donor agents. And those were important because, you know, it's a lot easier than working with NO gas. If you work with NO gas, you've got to go to the hood. You use, you know, a microliter gas-tight syringes. You have to mix NO with argon or oxygen-free nitrogen and then make dilutions. Very difficult to work with, whereas an NO donor chemical is a lot easier to work with. Now, S-nitroso-N-acetyl-penicillamine, which I have there, was a stable crystalline compound. The others are all unstable. You have to make it, put it in methanol, and use it right away. But SNAP was quite stable, very easy to work with. And so these experiments actually represented the very first uses of S-nitrosothiols as NO donors in biology. And a few years later, after publishing work on SNAP, good old Sigma Chemical Company made it widely available to researchers. So that was fun because you didn't have to work with NO gas anymore. And during this, we looked at platelets because we found that, I'm going to try to get this to work, sorry, there we go. We noted in the literature that sodium nitroprusside, which as you know is a vasodilator and had been used as a vasodilator, was also noted to prolong prothrombin bleeding time. And it worked by inhibiting platelet aggregation, but nobody knew how. Well, we knew that nitroprusside relaxed vascular smooth muscle because it was an NO donor, released the nitric oxide. So we thought, well, maybe that's how nitroprusside inhibits platelet aggregation. And so we decided to test nitric oxide on human platelets to see if NO would inhibit platelet aggregation. And that's exactly what we found. And we published that in the journal Blood and a series of other journals. And as expected, the NO worked by a cyclic GMP-dependent mechanism. Now, we discovered this in 1980. And these findings, as well as many subsequent studies by others, explain the protective action of endothelium-derived nitric oxide in stroke and heart attack and similar disorders. Now, we also turn to the mechanism of activation of this cytosolic guanylate cyclase by nitric oxide. And of all the basic research projects that I've ever been involved with, in my research career, this one was the most stimulating, but the most challenging. And one of the most challenging steps was to reproducibly purify this enzyme to homogeneity. And here's an example of some of my frustrations. Our first successful enzyme purification resulted in the elution of a concentrated homogeneous protein solution that was red in color. Bright red, you could see the color. And we determined that the red color was due to the presence of heme and heme iron. This was originally suggested by several groups, and we went on to show this directly. But during our second purification attempt, the enzyme protein, although it eluted as a single band, it was homogeneous, it had no red color. Solution was crystal clear. Why was that? Well, I don't know, but maybe because we had a technical problem in the cold room in that the compressor broke down during the last purification and concentration step. The temperature had risen to 22 degrees centigrade, which is not good for proteins. And so upon conducting our standard guanylate cyclase assay on the prep, we noted that the basal or unactivated catalytic activity was just the same as the enzyme that had the red color, but it could not be activated, not one bit, by nitric oxide. And so we thought, well, why was this? Could it be that heme was required for enzyme activation by NO, and that somehow the heme dissociated from the enzyme protein at elevated temperatures in the malfunctioning cold room? And the answer to both questions was yes and yes. So when heme was added back to the heme-free enzyme in the assay tubes, it was totally activated by nitric oxide. And we determined using gel filtration experiments and other experiments that the heme actually bound stoichiometrically to the guanylate cyclase. So our conclusions from these experiments were twofold. One, if your cold room breaks down, don't throw away your purified enzyme proteins just yet. And the second conclusion was that heme is required for guanylate cyclase activation by NO. And now, of course, we know that to be the case. So let's go back a few years to about 1980, which is about 4 years after the discovery of NO, at which time the pharmacology of NO was certainly fascinating, and this was the pharmacology at the time. Again, 1980, it was known to be a vascular, a non-vascular smooth muscle relaxant, and this we knew just prior to the famous discovery by Robert Furchgott of EDRF in 1980. So NO is a vascular, non-vascular smooth muscle relaxant. In vivo, using NO donor drugs, it lowers the blood pressure, it's a vasodilator, improves local or regional blood flow, inhibits blood clotting, as I told you, by interfering with platelet aggregation, and all these effects are mediated by cyclic GMP. Okay, well, that was great. Very nice pharmacology, but something was missing. What was missing? What is the physiological relevance of all of this? Physiology. What's the physiological relevance of NO, which is present in the Earth's atmosphere? So what's the physiological relevance of the NO cyclic GMP pathway? We have cyclic GMP in cells. Okay, fine, great, it's inside the cell. Can activate guanylate cyclase, it goes up. Where's the NO coming from? The outside? And so we thought, that's what I mean, nobody was thinking about nitric oxide as a naturally occurring signaling molecule in cells because of its reactivity with oxygen and formation of NO2. If you've worked with NO in the lab, you know that it's going to steer you away from thinking that NO could exist in cells, but not so fast. You know, we would ask ourselves at our Friday afternoon laboratory meetings, why would a pure exogenous molecule like NO be such a potent vasodilator? It actually works in the picomolar range. Why would well-defined receptors exist for NO in cells? What are those receptors? The heme groups of guanylate cyclase. So is it possible that NO does exist in cells, but we just didn't know it at the time? Could this explain the physiological relevance of the NO cyclic GMP system? Well, so we made a decision to look for NO in biological tissues, starting with arteries and veins. And it was difficult to do. But as we were doing this, I read more and more about the chemistry of nitric oxide, and my conclusion during this reading was that who would ever reason that NO could exist endogenously in mammals, let alone function as a signaling molecule? And the more chemistry you knew, the more convinced you were that NO could never operate as any kind of a signaling molecule, and here's why. Let's look at one chemical property. There we go. It's a small lipid-soluble molecule, okay, diffuses readily through membranes. It can't be stored in any organelles because it diffuses through these membranes. And so, back then when we thought about signaling molecules and neurotransmitters, we thought of all this organized, storage and release and formation and so on. So how could NO function, as any kind of a signaling molecule? It's a reactive free radical with a very short half-life, reacts with paramagnetic species, including oxygen. So how could a signaling molecule exist like that? It would be readily destroyed as soon as it were introduced or made. Not only that, it binds to iron, it binds to sulfur, it binds to other elements. And so we concluded that NO simply could not exist in vivo, and we lost our enthusiasm to hunt for NO in mammalian tissues. But then something remarkable happened. Bob Furchgott discovered EDRF, or endothelium-derived relaxing factor, which explained, of course, the vasodilator mechanism of acetylcholine, which, by the way, as you know, had not been known for 100 years since the first experiments with acetylcholine. And so the following animated cartoon series illustrates the progression of my own logical and personal thoughts that culminated in our discovery that EDRF is nitric oxide. And so I'm going to take you through this chronologically, and this was good for me to go back and look at everything I did chronologically and kind of put it together. And pay attention, because I'm going to ask you a question as soon as I'm done with this. So this is what Bob Furchgott first discovered. You know this better than I do. So here we have, this could be an isolated arterial ring or a helical strip, carefully prepared so that the endothelium is intact. And this would be sort of like the lumen out here. This is an isolated preparation in a tissue bath. We have many layers of smooth muscle. I'm just showing one here. This is the endothelial cell layer, and as you know, that's a single layer of cells, not multiple layers. So what Bob found, very simply, is that acetylcholine, by interacting with muscarinic receptors on the endothelial cells, generates something, a relaxing factor, which he didn't know what it was, called it EDRF, endothelium-derived relaxing factor, and somehow this interacted with the smooth muscle to cause relaxation. And so, we had talked about this, I remember, and I asked him, well, what is it? What do you think it is? He didn't know. Nobody had any idea at the time what the chemical nature of EDRF was. And so this is the way we sort of did this. We knew that cyclic GMP and cyclic AMP, the two cyclic nucleotides, cyclic G and cyclic A, were vascular smooth muscle relaxants. So we wanted to determine if maybe it was possible that when acetylcholine was added to this preparation, cyclic GMP or cyclic AMP or both went up. And when we did the experiment, we found that only cyclic GMP went up. Cyclic AMP levels were unaffected. Now, the endothelium dependence of this effect suggested, if not indicated, that EDRF was responsible for elevating the cyclic GMP levels. And so, therefore, we asked the next question, and that is, was cyclic GMP here required for the relaxation, or did it go up for some other reason and has nothing to do with relaxation? And so we tried to block guanylate cyclase by using methylene blue, which was shown earlier by Fred Murad to inhibit guanylate cyclase. And when we added the methylene blue, we blocked the cyclic GMP accumulation as well as the relaxation. So this indicated that, in fact, EDRF was activating or elevating cyclic GMP by activating guanylate cyclase, and the cyclic GMP was causing relaxation. And so, of course, the observations implied that guanylate cyclase was residing in these cells, but we wanted to make sure of that, and we wanted to determine if the EDRF itself was activating the guanylate cyclase. So we did those experiments, and we found guanylate cyclase in the cells, and that EDRF, these were tough experiments to do, very complicated, We published this, I think, in 1986. But EDRF activates guanylate cyclase. All this is endothelium dependent, and the cyclic GMP causes the relaxation. So now, at the time, the only, EDRF is a very labile substance. It has a half-life of a few seconds or less, depending upon the physiological or the bath conditions and so on. So at the time, the only labile substance known, at least to me, that could activate guanylate cyclase and stimulate cyclic GMP production was nitric oxide. And so if you add nitric oxide to this preparation, NO being very lipid soluble, just goes right through the membranes directly to guanylate cyclase, activates it to elevate cyclic GMP and cause relaxation. Obviously, the endothelium is not required for this effect. It's endothelium independent. So here's the question that I promised you that I would ask you. So if you look at this data that's right in front of you, what should you conclude? Take a look at everything, and what would you conclude? And if I ask you what EDRF is, what would you say that it might be? Well, it was very clear to us that EDRF and NO were indistinguishable, at least pharmacologically. The only conclusion we could reach was that EDRF must be nitric oxide. However, before we could publish such a provocative conclusion, we had to prove it. We had to isolate and identify the EDRF as NO. So in a series of very tedious experiments, these involved pharmacological experiments, physiological experiments, and importantly, chemical experiments, we did demonstrate unequivocally that EDRF is NO. And so now we could change the EDRF to NO, and now, of course, we understand the physiological relevance of nitric oxide and the nitric oxide cyclic GMP system. I always say that if Bob Furchgott had not discovered EDRF, we might still not know if nitric oxide exists in cells. And so we completed these experiments in November, December of 1985, and in fact, I presented these results that you see at a vascular biology conference at the Mayo Clinic on June 11th, 1986. I forget dates, but some dates one never forgets. And Bob Furchgott was there as well. In fact, he gave a talk before me. So we talked about this quite a bit at that particular meeting. And we were all very delighted because it was really the first demonstration that mammalian cells could produce nitric oxide. And then this field was advanced the next few months by contributions from Salvador Moncada and others. David Brett and Saul Snyder from Johns Hopkins showed they showed the existence of the enzyme that makes NO, that is NO synthase. And the first clinical application of nitric oxide gas came about a year after that by Warren Zapol at the Massachusetts General Hospital, where he pioneered the use of INO, or inhaled nitric oxide, for the treatment of persistent pulmonary hypertension in infants and newborns. And so much for the earlier supposition that NO could never function as an endogenous signaling molecule. I mean, you've heard people say never say never. And still today, I use the word never a lot and often prove myself to be wrong. And ironically, the very reasons that we chose not even to look for NO in cells, because it was impossible for NO to exist as an endogenous molecule, show us that these are all great reasons why NO is the perfect signaling molecule. It's a lipid-soluble substance, diffuses through membranes, it's made, produces effect right away. At distant sites, it doesn't care what membranes are in front of it, just looks for the heme group of guanylate cyclase. It's a reactive free radical. That's good. You don't want a signaling molecule to last for seconds or minutes. Let it last for a few, a tenth of a second, 1 or 2 seconds, and it'll trigger something else, and that something else will go on downstream to produce its effects. Okay, it reacts with iron. Sure it does. iron of heme is the receptor for nitric oxide on the guanylate cyclase. And yes, NO reacts with sulfur species. NO reacts with sulfhydryl groups covalently to modify the function of so many proteins that have sulfhydryl groups at the active site. And so this molecule, which we thought could never exist as an endogenous molecule, turns out to be a very widespread signaling molecule in mammalian cells. I always find this fascinating when I go back and look at all the reasons why I thought we should not look for nitric oxide. So finally, no talk about nitric oxide, at least given by me, is complete without a discussion on penile erection. I just threw that at you. And so moving onward and upward, let me explain that to you. We discovered that NO, I went through that quickly. So we discovered that NO is the long sought after peripheral neurotransmitter that promotes erectile function. Now, what possessed us to even think about this? I'm not a urologist. I don't even have an MD. I'm not a neuroscientist. I know very little about the brain, and I'm ashamed of that, but that's the way it is. But I was aware that the neurotransmitter released from the peripheral nerves that innervate the corpus cavernosum in the erectile tissue was unknown in the year 1990. There were no drugs to treat erectile dysfunction, which afflicts over 10% of the male population of the world. 10%. Amazing. And what caught my attention, I mean, the only things that were available in 1990 or before were certain surgical procedures. And I'm not going to discuss any of that with you today. So what caught my attention was that NO had just been shown to be a neurotransmitter of non-adrenergic, non-cholinergic nerves, as they were called then, the NANK neurons in the brain. This elegant work was done by John Garthwaite in England, and he thought that NO had something to do with memory and learning, postsynaptic effects, and so on. And then I knew that the nerves, so when he found that, I thought, why couldn't NO be a neurotransmitter in the peripheral NANK nerves as well, not just central NANK nerves? You see, the nerves innervating the corpus cavernosum were characterized by others as NANK nerves. And so we did the appropriate experiments using erectile tissue, first from rabbits, then from humans, and the rest is history. I'm not going to describe those to you. And we published our first paper in 1990 using rabbits, which was rejected, by the way, by Nature. And then we published our human study, which was also rejected by a pharmacology journal. But we got it published in the New England Journal of Medicine, which is the only paper I ever had in the New England Journal of Medicine. And that's shown here on this next slide. January 9th, 1992. And you know, many of you probably know that this journal is linked to all the news media. They're always looking for what papers are coming out. They actually get to see the papers the day before the lay public can see these publications. And so that morning, very early in the morning, I was bombarded with telephone calls from the news media, magazines, and so on. And I remember the first phone call I got was from the editor of Hustler magazine. It's 5:30 in the morning. I'm just drinking my first cup of coffee. And I couldn't decide whether to accept this interview or not. And then I remembered that my mom was alive and doing very well at that time. You know, she was not formally educated, born in Italy. She was very proud of her son. The Nobel had not come along. This is 1992. And I figured, what in the world is she going to think of her son if she knows that I'm interviewing with Hustler magazine? I didn't even know if she knew what Hustler magazine was in the first place. But then the second interview came from the New York Times. I accepted that interview, and the results of that interview were published in the New York Times when President Bush Sr. Was in office. And here's our article. Other than calling nitric oxide nitric acid, I thought it was a pretty decent article. And a similar article came out in an Italian newspaper, I'm Italian. I was born in Brooklyn, hey, and my parents were born in Italy. But in the Italian newspaper, I can tell you that sometimes Italians think better in using pictures instead of words. So in the article that they came out with in the Italian newspaper is a man in bed with his lady, and he's wearing a nitric oxide gas tank where the nitric oxide is written in Italian. So my mom did see this, and she was very disappointed. And just a couple of months after that New England Journal paper came out, the journal Science saw fit to declare nitric oxide as molecule of the year. So what happened 6 years later? That paper was in 92. 6 years later is 1998. What happened? Well, sildenafil was marketed by Pfizer Pharmaceuticals as Viagra. And of course, this works by increasing the action of cyclic GMP and nitric oxide. It's a phosphodiesterase inhibitor. It inhibits the breakdown of cyclic GMP so that if you have even a little bit of NO present, the cyclic GMP will go up tremendously because it's not metabolized by phosphodiesterase. Viagra inhibits that. That's how Viagra works. But of course, you can't get any increase in cyclic GMP unless you have NO around. So we like to say that Viagra increases the action of nitric oxide, although it's indirect. So let's look at the dates more carefully. It was March of 98 that Viagra was marketed. And what happens just a few months later? In October of 98, the Nobel Prize for nitric oxide gets announced. Was that a coincidence or what? So I decided to look at the Nobel Committee members for physiology or medicine. I looked them up, and I found that the majority of them were men over the age of 60. It's amazing how things work, but we'll take it. And so this is the poster. For every Nobel Prize awarded in every category, the Nobel Foundation has incredible artists who put together a poster. Many of you have seen the poster a million times, so I'm not going to go over it with you. But the three of us here sharing the prize, here's a heart in the middle, coronary arteries left and right. To the left is a healthy coronary artery making lots of nitric oxide. It's not inflamed. There's no atherosclerotic plaques. Things are fine. The lumen is got lots of room to traverse the blood. And on the right side, we have an artery that's pretty sick, probably from someone who had diabetes or somebody not leading a healthy lifestyle, a sedentary lifestyle, poor diet. At any rate, very low nitric oxide levels. This results in the deposition of atherosclerotic cholesterol plaques, results in proliferation of vascular smooth muscle because NO by itself actually inhibits vascular smooth muscle cell growth. So when you lower the NO, you get enhanced growth, and that occludes the lumen even further. And what's incredible, which you can't see on the slide, but you can't see in the posters all over the place in this downstate medical center, is the 3 experiments that each of us did that the Nobel Committee thought, we deserved the Nobel Prize because of these experiments. So I thought it was really very, very nicely done. And so on the few slides that remain, October 12th was the lucky day. That was Columbus Day, but a few years later. And this is when the Nobel Prize for nitric oxide was announced. And I was not home at the time. I didn't know the Nobel Prize was going to be announced. I mean, I knew it was going to be announced, but I didn't think it would be, for us. And I wasn't sure what date it would be announced. I had accepted an invitation to go to Italy, Naples, Italy, where my father was born in a small town out in the Naples suburbs here. He passed away many years before this announcement. But I was here because I was invited to give a lecture, so I stayed. I gave my lecture. I stayed for 2 days. That got my dean and chairman and chancellor very upset. They wanted me to come home immediately because they had things planned. So here I am. The first celebration was with my medical students, who I love to teach. They were so delighted that their nice guy professor came back with an announcement of a Nobel Prize and thought maybe I would skip the final exam because of that. And here, Sharon and I, my wife Sharon, she's an anesthesiologist. We're waiting in the Red Room to meet the President of the United States at the time in 1998. All American Nobel laureates are invited to go meet with the President. And there I am meeting with Bill Clinton at this time, and turned out to be a very charming individual. And here's all of us, all the American Nobel laureates. I'll just point out Bob here, and this is Fred, this is myself, physics here, and chemistry here. This is Bill Clinton. And you look at the smiles. Look at the smiles in our faces. We were so pleased to be there. Do you think if we got awarded the Nobel Prize this year, we'd have those smiles? I don't think so. And anyway, here's Sharon and I, a photo with Bill Clinton. This was really a great moment. And I remember when this photograph was taken, I could see Hillary Clinton walking in the distance, and I kind of followed her around. I wanted to see where she was going because I wanted to catch up with her to continue a conversation that we had started previously. And then I turned back around to look at my wife, and this is what I see. Some of you, I'm sure, remember this man. I can tell you from personal experience that this guy is faster than the speed of light. And what's even more disconcerting about his speed is the expression on my wife's face. And then we're off to Stockholm just a few weeks later, first week in December, where we all stay at the Grand Hotel. This was a photo I took in the summer because in the winter it's all covered with snow. And this is the venue, which you've seen, all over here. So in the first row are the Nobel laureates for this particular year in all the categories except peace. The Peace Prize is given out on the same day, the same hour, but in nearby Oslo, Norway. That's what Alfred Nobel wanted. And so behind us are seated prior Nobel laureates, other distinguished scientists and guests. And on the right side are members of the Nobel Committee for each of the categories, as well as members of the Nobel Foundation. And here we are back to the slide. Here's Bob Furchgott, myself, Fred, the three individuals who got the prize in physics, John Popel in chemistry, Saramago in literature, Shen in economic sciences. And this is during the day of our lectures. Here's Bob with his briefcase, as he always had with him. He was getting ready to give his lecture. Each of us gave a lecture, a 1-hour lecture, and he went first. He was getting ready to give his lecture at this meeting. And this slide actually is the day after the Nobel ceremonies. This is when the Nobel laureates and their spouses spend a very special day with the king and queen in the castle, in the nearby castle. And we take photographs and drink brandy and smoke cigars, if you're into that. And here's Margaret and Bob, Carol and Fred, and Sharon and myself with King Carl Gustav and Queen Sylvia. And of course, here is Professor Robert Furchgott receiving the Nobel Prize from King Carl Gustav. And look at that face. There is one very proud man, well-deserved, of course. And here he is. We asked him to give the banquet. At the banquet, one of the three Nobel laureates, or just one, gives the banquet speech, welcome speech, few comments. He did a very nice job with that. And here I am, I have to show me, right? Here I am getting the Nobel Prize from King Carl Gustav, and I have the same kind of proud face. This is a hell of a moment where you've spent your entire career working and wondering what you're doing with your life, and then finally you're on this stage receiving the Nobel Prize. It's unbelievable. But what I want you to remember is not so much my getting the prize, but what I want you to remember is the importance of nitric oxide. You can never get away from nitric oxide, ever. I'm going to stop here, and I want to thank you so much for your attention. Thank you. I appreciate that, thanks. And thanks for coming.
Does anybody have, we have time for maybe a question or two, if anyone has time, but otherwise we can.
I'll be outside if you can
Be
ask me
outside,
there.
you can mingle, you see the wonderful gentleman, full of stories. And I also encourage all of you to go take a walk through the gallery. You can see Dr. Furchgott's handwritten notes where he asks himself, why not NO? Why not NO? Right?
Exactly.
So please, thank you for coming. We really appreciate your support of our society. And Dr. Ignarro will join us all outside.